All right, so we just wanted to discuss, give an overview over this LCCMR product, with LCCMR grant, sorry, which is focused on the biocontrol of Japanese beetle with the intention of preserving pollinators in natural areas, parks, and other places. So I wanted to just start out talking about the information that the Krishik Lab has available online, which talks about a range of different topics, from what bee lawns are to identifying different pollinators, and also this particular research project, and it's just there essentially to inform the public, and so that it's accessible to the advisory board to learn about the progress of this project and sort of the different management options that are available. This other site, which the URL is listed down here, is more specific to this particular grant. And so you can really look over the grant materials, which include our hypotheses about the research, what our objectives are, as well as a summary of the grant activity, which will be updated as we progress through the project. This is a list of all the participants in the advisory board for this project. So you can see that we have cooperators that encompass a range of different organizations. And so we have several members of the Minnesota Department of Agriculture, Minneapolis Parks and Rec Board, DNR, but also we have other participants who work in different capacities with the public, seed companies and master gardeners. And so we really wanted to assemble a group that sort of span this range of different stakeholders that are relevant to this topic, from those that work in parks and deal with using different management techniques for natural areas, or even people who just work with private homeowners and deal with sort of the management options that they might be looking for. This slide shows kind of a summary of the grant progress so far. So LCCMR does mandate that we submit a progress report every 6 months, which we will also send out to the advisory board so that you all have the information about what's been going on. This will be in addition, of course, to the Zoom meetings that we're going to be having about twice a year over the granting period. This grant the funds for the grant were received last fall in September, which of course was too late to really do any field work. And so the time spent last fall was really spent on updating the websites and making sure that we had all the information available to the public so that people could access information on integrated pest management and learn about pollinator conservations, and particularly in reference to parks and green spaces and bee lawns. I was hired in February, and so the work that I did over the spring was really to work with our collaborators at Michigan State to learn the technique that they had developed for identifying the pathogen in beetle samples and also in soil samples. And this is the analysis that we're going to use in our work for the beetles that we sample from the different sites. And it consists of a qPCR technique. So it's a technique that can measure, it uses primers for the pathogen, so it can measure the presence and also quantify the pathogen in beetle DNA extracts. And Dave Smitley's lab at Michigan has really pioneered this project. They've done all the work with the analysis and relating it back to infection rates that are diagnosed microscopically, looking for spores in beetles, which takes a really long time, which is why they developed this technique. And so we're going to work with them to use this technique to measure infection rates across different sites. Cody was hired in July. He started in July, as a researcher 3 to really conduct these toxicological bioassays for microbial products that are available for Japanese beetle management. And so he's been doing these dose response bioassays. He's also been supervising the sampling of beetles at over 40 different sites, which we are also doing with two of our undergrads, Michael Angstrom and Kyle Murley. And so they've been putting up Japanese beetle traps at these different sites, and they've been sampling twice a month in July, which will continue in August and also in September. And these are the samples that we're going to be analyzing using this qPCR technique to determine infection rates in beetles at each of these different sites. In addition to this work, we also in the spring and beginning of the summer produced some publications. So I sent out an article, the Whole Notes article, which was published in the MGCSA magazine. And there's also an article that we published in the MNLA magazine, as well as a peer-reviewed manuscript that was really a review of microbial biocontrol for scarab pests that we're currently editing and working on submitting. And then sort of the final piece of this work is the outreach and education aspect. So we're working on developing these demonstration projects with the Minneapolis Parks and Recreation Board. And so this is to create signage that we can put out in two different locations in different Minneapolis parks, high traffic areas where the public can see them. They can learn about pollinator conservation, and they can learn about IPM management, including this project and the biocontrol agent that we're working on, just to spread awareness. We're also looking for an additional site, which we'll probably put up next year. So just a little bit more background of this project, and in particular Japanese beetle and why we're focusing on that. Japanese beetle is an invasive beetle from Asia. It's actually been in the United States for quite a while, however. Was first detected in 1916. It was commonly found in Minnesota in the 1990s, and it's sort of renowned for being this, really effective defoliator of different plants. So it feeds on, the adults feed on foliage and also fruits of different plants. And so in this capacity, they can also impact pollinators by reducing food resources for bees and other wildlife. So for the long-term research outcome that we would like to see for this project is to try and establish this pathogen called Ovovesicula papilliae as a viable biocontrol option for Japanese beetle. And this was first detected in Connecticut when they were doing a survey for scarab pathogens, and has since been introduced in 5 different states to try and establish the populations of this fungus in the soil. And particularly in Michigan, where our collaborators work, they've done a lot of work on this. Also in Colorado, they have quite a long data set on how the establishment of Ovovesicula impacts Japanese beetle populations. It infects the grubs in the soil, and it's primarily the mortality it causes is that it reduces overwintering survivorship of the grubs, and this could be reduced by as much as 50%. But in grubs that are infected and do survive to adulthood, it also acts to reduce fecundity by 50%. So even though infection occurs only in the grubs, it can have latent effects in adults. And it can decrease beetle populations, depending on data depend on sort of where you look, by 60%, in about a 5-year span of time. So that's sort of a short-term effects, but you get a very significant increase in control over a 20-year period. So the data set that's available show that you can get as much as 75% reduction in beetle populations over this long term. And so that's kind of what this pathogen is really going to be most useful for, is really long-term control. We have found the pathogen in Minnesota, so we know that it's here. We found it in Stillwater at a site and also on the St. Paul campus. But of course, a large part of this grant is to really expand this survey and try and see where else it might be located. We've also been, so in addition to this long-term management with the use of this ovovesicula, we're also focused on trying to provide information on short-term management. And so we're testing a range of EPA approved microbial products such as, BT Galleria, which is available for Japanese beetle control, as well as the fungal pathogen Bavaria bassiana, parasitic nematodes, and then bee-friendly insecticide, which is available that is safe for bees, although there are some complications with other types of pollinators. But it could be a viable, option for use for Japanese beetle control in some of these areas. And Cody's really spearheading this, so testing the efficacy of these products. And also, we're planning on testing combinations of these products too, to see if we can recommend use of dual, products that might be synergistic. And of course, this is a type of data that we really want to try to include in these demonstrations to alert the public to the existence of these products and the efficacy of them, to try and increase adoption of these bee-friendly products as alternatives to other insecticides that have non-target effects on pollinators. We did want to point out that in the past, there have been efforts to research other biocontrol agents for Japanese beetles. So there's a parasitic fly, Stochetta, and also a Typhia, which is a parasitic wasp that one lays eggs on the adults, the other lays its eggs on the grubs. And so these were found to be effective at, essentially killing Japanese beetle. And the MDA did do field releases on both of these species in the late 1990s, but found that establishment wasn't very high. And so they didn't really see significant reductions in Japanese beetle populations. We do know that, these species are out there. We will find beetles that we can see have eggs, on them, but it just doesn't seem to be an efficient enough control, biocontrol for populations. So because of this, we're really focused on testing this other biocontrol agent, ovovesicula. And as I said, we know it's here. And so we're trying to locate other places where it might be present. And eventually this would be in the goal of being able to release it at different sites and try and get it established in the soil, so that we could, really test its ability to control populations over time and essentially do what our collaborators in Michigan have done there at different sites. They have really good data on establishment and control. And as we mentioned, in Michigan, they found that, post-inoculation at some of these sites where they've tested it, you get about 30% reduction, but really over longer term, 18 years, they saw, a reduction by 75%. So there's a lot of promise for this at being a really good option for long-term control in some of these areas. As I mentioned, so a huge part of this project is to really disseminate the information we have on what's available short-term, and then educate people on this potential other product for long-term control, which we'll do through these demonstration projects that we're going to have set up in different parks, hopefully in very high traffic areas, to just educate consumers about not only about Japanese beetle, but also about pollinator identification and what bee lawns are, and why it's important to think about what you're using for insect control, even in private yards and things, because these pollinators are around and we want to make sure that we're not having non-target effects on them. So we're working with the Minneapolis Park and Rec Board at least 2 of these sites, and they're helping us with getting the signage set up. Essentially, it's going to be, look something like this. when we get them set up, and we might also have brochures available for people to take. The last thing I really wanted to talk about is some of the other resources that we have available through our lab. So there's a lot of information across these websites for information on IPM, for greenhouses and nurseries and ornamentals, but also on pollinator conservation and pollinator identification. And then, of course, for those that are interested in obtaining their pesticide certification, we have a lot of training materials that they can use for studying before they take their tests. So, you know, we wanted to just alert you all that these are other resources that might be useful for you and your organizations. So Barry, did you want to talk about this, or? You're muted. There you go.
Do you hear the birds in the background?
No,
Okay, good.
not today.
Thanks, Kara. That was an excellent summary of the research and outreach on this project. And so I just wanted to continue with a little bit more in depth for those of you who don't know that much about Japanese beetle. So the purpose of our meeting today is to let you know what we're doing and to know you can contact us if you need for your specific group any more information about Japanese beetle management. And also if you need some handouts or information, or in the winter, if you need somebody to give a talk, we're available. We also want to start thinking about getting a renewal for the grant, because we know we cannot do this work in 2 years. Dave Smitley started his project in 2005. He got money from the Rose Society in Michigan. They're facing the same issues that earlier Jim Calkins mentioned and Aaron Burkholtz from the Arboretum mentioned. And so there needs to be ways to manage Japanese beetle that are not just chemically dependent. So we are trying to set up a website where we have information for people based on the sites, whether they work at parks or green space, whether they're a consumer or they're an arborist or a landscaper, so you can get correct information. So we have about 10 to 15 bulletins we've done over the years. We also, since starting in about 2019, we also if you look on the banner, the gray banner at the top under pollinator and beneficials, we have a great website where we have identification of ground nesting wasps, ground nesting bees. As a matter of fact, all the families of wasps and bees in Minnesota, there's about 350 species of native bees. And people need to know this when they're managing landscapes, how to tell the difference between a wasp nest in the ground and a ground nesting bee. So for instance, Erin at the Arboretum just sent an email on Friday. She had ground nesting bees and she wanted to know somebody was stinging kids at the Arboretum, and she wanted to know if it possibly could be these ground nesting bees. And so we talked about it. We looked up what other people have said and my own experiences. And we said, no, those are not the culprits. You can get really close and photograph them. So you can leave them alone, let biocontrol take its course. This happened to be a ground nesting wasp that killed marmorated stink bugs. So we don't want to get rid of that biocontrol agent. So it's amazing different sites, how you need site specific information. And that's what we've been developing on this website. Next page, please. So we just wanted to go back, and the reasons why we started on this grant was because of rusty patch bumblebee, which is an endangered species, and monarch, which this week the international unions of concerned scientists listed it as a potential endangered species. The U.S. Fish and Wildlife Service have not done so. So rusty patch bumblebees, you see in the horizontal lines there, that was its native range 20 years ago. Rusty patch bumblebee, yellow-banded bumblebee, eastern bumblebee, all were very common. And now all of them are disappearing. And those little dots are where active populations are. We have some populations in Minnesota that people are bumping into. There's this really good online app called Bumblebee ID, bumblebee.org, and you can send a picture. Landscapers can send a picture there And so what we need to do is develop site-specific management. When a landscaper comes across Rusty Patch Bumblebee, how does he work with the DNR, the Department of Ag, to create a management plan that is conserves Rusty Patch Bumblebee and monarch butterflies? So there is a lot of site-specific information on IPM that our website can help you with. Yeah, see at the bottom there, it says bumblebeewatch.org. Take a picture and send it for species confirmation. Next slide, please. So in this booklet called Butterfly Gardening that got an award from American Society of Horticultural Sciences last year, Lori Snyder, my previous lab manager, and myself wrote it. It is really excellent. It goes through all the behaviors of butterfly, overwintering sites. It has all the species. I wrote this bulletin originally in 1998, and this is a really big upgrade of it with wonderful pictures that Lori supplied. So please check it out. But on this particular page 14, we have pictures of threatened and endangered species in Minnesota. You see Rusty Patch Bumblebee, Carner Blue Butterfly, Yellow Banded Bumblebee, Monarch Butterflies, the skippers that the Arboretum works on. And then at the top, it tells you how to use IPM to manage your site-specific site. So site-specific IPM is really informational rich, and people need to work with somebody on creating an IPM program. Next slide, please. This is a poster we have. It is on Think IPM. Again, it was done by Lori and myself back in 2020. We have 3 other posters. They're available for downloading. So if you want people in your group to think more about IPM, just download these and print them up. And it goes into the 2 basic issues now that I'm going to talk to you about. What pesticides do you use to control biocontrol? So up there at the top, it says biocontrol, and underneath it towards the bottom, it says chemical control. This is the big issue of what chemicals do you use? And this is actually the second part of our grant that Carrie talked to you about, is in short-term management. What Cody is working on is trying to figure out what microbial insecticides do work. They can replace the conventional insecticides, and they can manage or conserve these biocontrol agents. Next slide, please. So here's just a picture of the butterfly gardening. You can download it from the website, the pollinator website. Next slide, please. So what is IPM? IPM is a system that is very site-specific. It's a decision-making process using multiple tactics. It's information-based, it's biologically based, it must be cost-effective, it's site-specific, and it goes all the way from regulatory aspects of Japanese beetle. How do you manage it at the airport when you don't want to ship Japanese beetle to sites around the country that have a quarantine, down to specifically in bee lawns, how do I manage Japanese beetle so I don't kill pollinators in bee lawns? Next slide, please. So biocontrol, here you see at the bottom an aureus. It's a species that's in nature. You can buy it from insectaries. At the end along that long thin tube, which is its beak, it's killing a thrips, which is the most difficult pest to manage in greenhouses. So biocontrol of conserving pollinators, conserving beneficial insects, conserving biocontrol organisms is all the same. You have to use a pesticide that conserves them, that doesn't kill them. You can't use the traditional broad spectrum pesticides. So these, you can purchase them, release them in greenhouse and nurseries. You have to release them at low population densities, because once you see spider mite webbing, they're not going to be effective. Next slide, please. So this is just a slide to make you understand how we are trying to get people not to use systemic insecticides, ones that you apply to the soil and get distributed around the plant. Remember, those all get into pollen and nectar and affect any insect. 99% of all the insects are good insects, and they need pollen and nectar. And so if you use something systemic, it's even going to show up in the extrafloral nectaries. So a lot of our grant is convincing people to use biorational, not organic, but biorational pesticides that manage pest insects, that have a limited duration, you spray it, and sunlight or microbes denature it. Next slide, please. So it's just some examples: acelephrin, chlorothan, and nitropol. You can use in grubs in the soil or on landscape plants as a foliar or a granular to get into turf, to kill Japanese beetle grubs. It is very bee friendly. And you'll see we're going to go over a bulletin at the end of this talk where you'll see how friendly it is to bees, but it kills monarch butterflies. So you don't want to use it anywhere near monarch butterfly hosts. Spinosad is another biorational. It is also registered for organic production. It kills caterpillars and sawflies. It works so well. There is no reason to use a pyrethroid or a carbaryl or even a pyrethrin. Neem oil works very well on aphids. And sometimes you do need the systemics, the neonics. Imidacloprid is used in the emerald ash borer program. Our ash trees in Minnesota, they plant a seedless variety. It only produces male flowers that produce pollen, but there's a mite that gets into all those male flowers and it prevents them from producing pollen. So in either endomectin benzoate or any neonic that you use in the ash tree, it's going to be hard for me to know how it's going to kill pollinators. However, you don't want to use a systemic insecticide on linden trees, and Japanese beetles do like linden trees. Next slide, please. So here's the distribution of Japanese beetle. It showed up in 1917 at an arboretum in New York. It got to Minnesota in 1990, but you need to recognize there are other states that have a quarantine against them, and we don't want to be shipping it from Minnesota to these quarantine states. Next slide, please. Japanese beetle adults feed between the leaves, the leaf veins that we call the skeletonizing. They're avoiding the thicker veins. Next slide, please. But they do produce a pheromone that causes them to aggregate. But even more important is that when they feed on the plant, there's these plant volatiles that are released, and that attracts them to each other. And so you see things like this in gardens and parks. And these Japanese beetle aggregate, they go to the top of the plants in the sun. And then when the night comes and it's dark, photosynthesis stops, and the plant stops producing those pheromones, and both males and females fly to turf to lay eggs. Next slide, please. And so here you see a linden tree, July 8th, July 18th, severe defoliation by Japanese beetle. Next slide. Here is the raster of a Japanese beetle, which is used to identify Japanese beetles. There are different sutras and hairs. Here you have a picture of the different ones for different beetles, so people can clearly identify the different species in turf. Next slide. But I have to say in Minnesota, although you have these 2, 4, 6, there's about 8 species total, 2 aren't on here. The major pest is Japanese beetle. Everything that we're looking at that is for biorational control would affect Japanese beetle. And let's say European chafer. There's an article in the Star Tribune. This might become a pest. It is a pest in Michigan. So far, it isn't here. So we are making sure that we include pests treatments for that pest as well. And Michigan is actually trying to sort out whether oviposicular will affect European chafer as well. Next slide, please. So Japanese beetle adults come out in the first week of July. The MDA as well as myself trapped for years, and it's pretty much guaranteed. They are around September 15th, and then they slowly taper off towards the end of September. Next slide, please. So this is what, here's 2 mating beetles, but what I want you to see is those 5 tufts of hair that make it recognizable as a Japanese beetle. That's the grub in the soil that you see as well. Next slide, please. So if you have Japanese beetle problems, the turf pulls up. There's no roots. Now on the left, you see turf that could be Japanese beetle, but a lot of the time in Minnesota, that's a fungus. And for the fungus, you need very specific fungicides. So I think people end up spraying more insecticides for Japanese beetle that isn't necessary because they think it's the beetles, but it's a fungus. Japanese beetle females go to turf at night. That's beautiful blue color. They don't go to turf this color, yellow. That means it's stressed. So in Minnesota, because we have this drought in July and August, they're really going mostly to golf courses, municipal lawns that are irrigated. The females don't like to oviposit in dry soils. Next slide, please. So JB control, actually the standard insecticides that you would use in Minnesota, carbaryl or Dilux, they don't work well. Carbaryl doesn't penetrate down to the roots. Dilux doesn't work well in Minnesota soil. Aceleprint is bee safe, but not butterfly safe. So in actuality, a lot of the conventional insecticides that people work are useless for controlling Japanese beetle larvae and adults. And so we really need to change our mindset and switch to these microbial insecticides. Next. Here's just a list of some of the systemics and common foliar. Remember, chloropyrifos is still allowed in golf courses and landscape use. All of these are highly toxic to bees. So the EPA has a system where they rate toxicity, moderate, slightly, non-toxic, highly toxic, and these are all highly toxic. Next slide, please. So just remember what I just said. Acelepryn gets on a plant leaf. It's around for 4 weeks. It's hygienic, is OMR approved, Organic Materials Research Institute. Acelepryn is safe for bees. Hygienic isn't. Hygienic has pyrethrins in it, but it has piponryl butoxide, and that will kill everything. So even though it's a pyrethrin, and even though it says OMRI approved, it is not bee friendly. Acelepryn is bee friendly. So any of the pyrethroids, pyrethrins, piponryl butoxide, PBOs out there, all of these, including carbaryl, are going to kill bees or even and monarchs with foliar applications. So what our grant, next slide, please. What our grant is trying to do is convince people to use these microbial insecticides and acelepryn, active ingredient chlorophenolnitropol. Scott's GrubX contains it, which is great. Scott's has been very proactive with its lawn controls, its lawn insecticides. Before acelepryn, GrubX had halofenicide, which is an insect growth regulator, which stopped grubs from developing. Acelepryn's had GrubX in there. GrubX has had acelepryn in there for at least 5 or 6 years. And another thing we have to educate consumers is that they know neonicotinoids are bad, but they don't know there's different active ingredients with different trade names. So their landscaper may say, oh, I'm going to use Arena, Merit, Meridian, Xylom. I'm going to use dinotefuran, thiamethoxam, clothianidin, and they don't realize that all are neonics. So there has to be education that maybe what you think or you're using is bee friendly is not bee friendly. Next slide, please. And so this is what we want people to change to. Bacillus thuringiensis Galleria BTG. It kills most of the scarab beetles in the ground. It's a new product. Recent research has even showed that it's effective as a foliar insecticide against Japanese beetle. Next slide, please. Nematodes, this is a picture from our lab. An elm leaf beetle, you spray it, you purchase the nematodes, they're formulated in powdered milk, you hydrate them, you look under the microscope or with a hand lens, you see them wiggling, you spray it, and you put 1 nematode into an elm leaf beetle, and this is how many you get out. But the issue with the parasitic nematodes is you really need moist turf. It works well in greenhouses, but in the dry Minnesota landscapes, it does not work well. Next slide, please. So I'm going to end with this slide. These are Japanese beetle lures, 2 different kinds of traps on each side, on the left and right. And then in the middle, you see the lure, that kind of aluminum foil. The round circle has a smell of roses. Germano is the active ingredient. And that little orange scepter is a rubber that's been impregnated with the pheromone. And these work really, really well, calling in thousands of beetles. And so one of the things we're going to do in our lab, Cody, the lab researcher, lab manager, is going to look at whether we can use traps, Japanese beetle traps, put BTG in it, put Bavaria, which are 2 fungal microbial insecticides that work against Japanese beetle adults and grubs, whether we can put it in there and disseminate it. So if you put it on the beetles, they don't die. They're full of the spores of both of those different pathogens. They fly back to the turf. They release it, it gets in the eggs or the grubs or the insects in the turf. This is much better use than spraying the insecticides with the BTG or the Bavaria. Because when you're spraying, you might accidentally hit a bee or a butterfly larvae. But if you're having the beetles have their bodies coated and they're disseminating it, it really does reduce risk. So this is what our grant is all about. Carrie did a great job introducing it to you, talking about why we want to do this, both the long-term issues of getting over vesicula more established in Minnesota. I've been trapping for the last 5 years. We know it's here. We trapped in 4 sites. We know in 2 of the sites it's common. And so 50% of the sites we looked at have ovovesicula. So I just got an email from Lori, and she wanted to talk about chickens in these traps. And when I first started trapping, I went to this small rose, landscaping rose site in Hastings. And the guy's been growing different rose cultivars for years. He's from Morocco, which is a big rose cultivating state for the European market. And I put out these traps. When I go back, I could never find Japanese beetles. And finally, he admitted to me his chickens spend the whole day underneath them eating beetles. And Erin, who was on before during this meeting, she talked about at the Arboretum, how the turkeys sit around the traps all day long. And I put an article in the Minnesota Golf Course Superintendents Association and actually had a picture. Organic growers set up these traps in blueberries, raspberries to pull the Japanese beetles out. And then they have guinea fowl or chickens. So yes, Lori, traps can be used to help chickens get a meal of Japanese beetle. So that's all I wanted to say with the slideshow. But Carrie, if you can help me out and just load that PDF of a bulletin that's available on all our websites.
This one?
Yes, can you see it?
Yes.
Okay. So this bulletin is something we've developed over the last few years. It's on the website, and it's for people to answer the question is, are the chemical that my managers are using, whether it's a consumer who has hired a landscaping firm or it's a municipality and their own ground crews, and you go through it and it uses the EPA toxicity to a B. So this is their standard method of comparing whether a pesticide, whether it's a fungicide or a herbicide. This is just an insecticide table. And it tells you how toxic it is. And there is a rating system. So you see the traditional thing you would use, foliar application for Japanese beetle adults or grub 7 is highly toxic to bees. So this over here, when it's to the left, it means it's usually used as a foliar application. These are something you put more usually in the soil. And again, these are the neonics by the different active ingredients. There's 4 or 5, and they're all very toxic. If we go down here to what we've been talking about, chlorothalonitropol, the active ingredient, the trade name is Celiprin. Look, it's greater than 104 micrograms per bee. So it's non-toxic. If you look up here at carbaryl, it's 0.014. So if we move these decimal points over, we have to move them over 6 spaces, which is 1 million. So cyanotronic chlorothalonitropol is 1 million times less toxic to bees. But remember, it is toxic to butterfly larvae, very toxic. So this table is essential. This is the only way you can work with your staff to sort out the chemical you're using. Is it bee friendly or not? So there's a whole group of miticides that are not bee friendly. Some are not bee friendly. There is lots of times the insect growth regulators that were used against pest insects. A lot of those are bee friendly. So again, putting this pesticide choice back in terms of biocontrol, and you're developing your site-specific IPM program, is we want people to use pesticides that not only conserve bees, but conserve biocontrol agents. So we hope today's meeting, Carrie did a stellar job talking to you about the grant. I gave you a little background information. We're hoping that you can contact us if you need a talk or if you want to change your management practices in the group you're in, so we can help conserve pollinators and use these EPA registered pesticides that control pests. A few years back, I got some money from the MNLA Minnesota Nursery Landscape to come up with what pesticide could replace the neonics in greenhouses. And I looked at this pesticide, pymetrozine. It's called Endeavor. And these two pesticides here, their mode of action is they prevent the siphon from working in an aphid, any insect with a siphon, with a sucking mouth part. Look at these high numbers. They're highly untoxic to bees. On the research I did, pymetrozine worked just as well as the neonics. So here would be your choice if you're a grower releasing biocontrol organisms, or if you're a grower selling plants that need to be installed in the landscape, is to use pymetrozine instead of the neonics. So that's our job here, both trying to establish ovovesicula for long-term management as a biocontrol agent for Japanese beetle, as well as coming up with better choices to conserve pollinators, beneficials, biocontrol agents in your short-term management programs. So thank you very much for attending. Please contact us if we can help you. Are there any questions? Lori, do you have a question?
Sorry. Well, that was all really good information. I would be curious to see the comparison, and maybe you can just talk about this, Vera, between turf that has been managed with ecologically sound practices compared to turf that's been treated with say a neonic or a systemic insecticide, if that has any effect on the health of the soil or the plants, for instance.
Well, Lori, I think that's, I am very into sustainable management, replacing conventional insecticide use. And the data is increasing not only in honeybees, that fungicides and herbicides affect the biome, the bacteria and fungus in honeybee guts, but that it's affecting the fungus in the soil. And so one of the things I sent some papers to our group was to look at the interaction between, if you use a herbicide or a fungicide on turf, is it going to affect the ovovesicula? And maybe that's what's reducing its spread in golf courses, which use a lot of herbicides and fungicides. So I agree with you. It's just really hard to find people to collect that data from. It's really hard for golf course managers to want to even release what pesticides they use. They're all afraid of some kind of negative issues from their clients or people in the neighborhood. So I agree that comparing somebody that did sustainable methods to conventional is really a good idea. I think you have to set it up yourself in order to collect the data you want. It would be nice if you could collect it naturally. So I just got an email from a group that's working, I think they're working in, is it Stillwater? No, they're working up in Mahatamidi in that area. And he wanted to know exactly what you're asking, if there's any data. If our lab would go up there and collect bumblebees and stuff and try to ascertain pre and post sustainability management to see if it changes. And we can't do that. We're all grant driven. It would be wrong for me to have people do stuff they're not being paid to do. But I think it's an interesting research question somebody should do, especially looking at these herbicides. I firmly believe the herbicides are having a role in affecting ecosystem process. Did that answer your question, Laura?
Yes, it did. Thank you very much.